Great. Hi, everyone. Welcome to the CCOM-OE seminar. We are happy to have Dr. Emma Cotter with us here today. Emma recently joined the Marine and Coastal Research Laboratory at the Pacific Northwest National Laboratory, where she is excited to work on new applications for marine renewable energy. Previously, she was a postdoctoral scholar at Woods Hole Oceanographic Institution, where she was a part of the Ocean Twilight project. Emma completed her PhD in mechanical engineering at the University of Washington in 2019, where she was an NSF graduate research fellow and developed methods and instrumentation platforms for environmental monitoring at marine renewable energy sites. Broadly, her research interests lie at the intersection of machine learning, ocean instrumentation, and marine renewable energy. Thank you so much for joining us, Emma Cotter, and you can go ahead and present.
Great, thank you. So thanks for that introduction. As Nicole said, I am now a research engineer at Pacific Northwest National Labs. I've been there for about a week at this point, so that might be the first time I've said that out loud. So today I'm actually going to be talking about work that I did as a postdoc at Woods Hole Oceanographic Institution. Specifically, my talk today is entitled Extracting Insight from Multi-Scale Data in the Ocean Twilight Zone. And what I've been asked to talk about today is two different instruments that we've been using to look at the ocean twilight zone, a broadband acoustic system and a holographic camera. So today I'm going to give you a little bit of background about what is the ocean twilight zone, why should we care about that, and then dive into some of the ways that we've been using these instruments to look at that. So first, what is the ocean twilight zone? Well, the twilight zone, or the mesopelagic, and you'll probably hear me use both phrases interchangeably, is the sort of second layer in the ocean here, about 200 to 1,000 meters deep. It's below the surface waters, the epipelagic zone. And the reason that we sort of nicknamed it the twilight zone is because while it's really quite dark, small amounts of light do still penetrate to these depths. And because of how remote and difficult to study it is, we know relatively little about this region of the ocean. But from what we do know, the mesopelagic is incredibly biodiverse. We have fish, we have gelatinous organisms like jellyfish and squid, zooplankton, and just an incredible amount of life. And many of these organisms tend to form what we call deep scattering layers. So these are relatively organism-dense layers in the water column where organisms tend to reside. And another thing that we know is that many of the organisms within these layers participate in a daily vertical migration, where they migrate up to the surface at night so that they can feed when visual predators aren't able to hunt them as easily, and then migrate back down in the morning. So this migration actually ends up being an incredibly important part of the ocean carbon cycle because we have organisms migrating up to the surface to feed and then migrating back down. And the fact that many of these organisms are quite small and relatively low trophic level makes them a really important part of the ocean food web. So this sort of ecosystem here plays a really important role in the ocean. So it's important, like I just told you, but there's this really big push to study the twilight zone right now. And the reason for that is because there's been, as we have increasing global population and concerns about global food shortages, there's been increased talk about starting to develop fisheries in the deep ocean. But if this is going to happen, we need to understand the role that these organisms play in order to understand if this can all be done sustainably, and if it can, how do we regulate that? So some of the big science questions that are driving the research on this topic are how much biomass is there, just how much life is down there, what fraction of the organisms are migrating, and how is this biomass distributed? Do we have more gelatinous organisms? Do we have more fish, etc.? So how do we do this? How do we determine how much life there is in the mesopelagic? Well, a common way to study the mesopelagic and fisheries in general is using active acoustics. So typically this is done using an echosounder looking down in the water column like this. And if you haven't worked with active acoustic data before, this is what we call an echogram. So the time of day is on the x-axis here, and depth is on the y-axis. This color axis here is called volume backscattering strength. So without getting into specifics, this is generally going to be higher where we see more numerous organisms or more acoustically reflective organisms. So to put it most simply, more fish, bigger fish. And so looking at this echogram here, you can see that this data is quite informative. We can see some of those characteristics that I described in the cartoon a few slides ago. This data was collected during a cruise in 2019, and we can see that the deep scattering layer, so where we have an abundance of organisms, is between about 400 and 500 meters deep. And we can see that daily vertical migration, where in the evening we have organisms migrating up to the surface and then back down in the morning. So this is really useful information, but actually going from this data to quantitative answers to those questions that I had on the previous slide is pretty complicated. And there are a few reasons for this. This metric here, volume backscattering strength, which we abbreviate SV, is a function of the number of organisms within the acoustic beam, the volume of the beam. So that's something that we can measure and know for our specific echo sounder that we're using. And the target strength, or the amount of sound scattered by each one of those organisms within the beam. So looking at this equation, you can see how if we can make good assumptions about how the organisms are scattering sound, we can come up with a good estimate of the number of organisms. And this has been done pretty extensively. This is how a lot of epipelagic, so fisheries in the surface waters, are measured. But doing this in the mesopelagic is a little bit more complicated. And there are a few reasons for this. The first is that for many fish, the most acoustically reflective part of the fish, or the part of the fish that contributes the most to that target strength, is the swim bladder. And this is a gas-filled organ that helps the fish regulate its buoyancy. However, as we know, pressure increases with depth. So the shape of that swim bladder is going to change, meaning that the target strength of the fish is going to change. So for the exact same fish, we might see a different target strength at 700 meters deep and at 300 meters deep. Second, we know that not all fish have swim bladders. And to make it even more complicated, some fish are born with swim bladders and lose them as they age. And we don't really have a great estimate of what fraction of mesopelagic fish have swim bladders. But we do know that fish without swim bladders scatter sound entirely differently. And then, of course, we're not just talking about fish. As I mentioned before, we have gelatinous organisms like squid, zooplankton that are contributing significantly to the ecology of this environment, and other organisms as well. And finally, for any organism, the way that it scatters sound, so its target strength, is going to be a function of the frequency that you're measuring at. And historically, a lot of measurements have been made at just one frequency or at several discrete frequencies. But for reasons that I'll come back to in a few slides, for a lot of mesopelagic organisms, 38 kilohertz, which is a frequency that's been used quite a bit for studying fisheries and has been used in some recent studies studying the mesopelagic, is really close to a point in that target strength frequency curve where we're really sensitive to frequency. So small changes will result in big changes in our target strength. Okay, so hopefully I've shown you that this is quite complicated. We need more information than we're able to get from the surface to understand the twilight zone. So rather than just measuring from the surface, the team at WHOI, before I started my postdoc, developed a system called Deep Sea. They can travel down into the mesopelagic to better understand these complicating factors. So Deep Sea can measure individual organisms up close and hopefully start to give us a better understanding of these complicating factors across many different size scales of different organisms. Broadband echosounders, a multi-beam sonar, a holographic camera, optical cameras, a current meter and environmental sensors, and an eDNA sampler. So you can see how this is giving us many different modes of information across many different scales. And because this is a cabled system, we're able to continuously record data from all of these sensors as it travels down into the mesopelagic. So this gives us a super rich and exciting data set. But it also presents some new challenges in terms of just the sheer volumes of data that we're able to collect and types of data that we haven't been able to collect before. So today I'm going to focus on some approaches that I've been working on to address that challenge of how do we start to process and wade through this data that we've collected with DeepC. And I'm going to focus on these approaches for just two of the subsystems on DeepC, the broadband echosounders and the holographic camera. And I'm going to address these separately. So I'm first just going to talk about the broadband echosounders. And when I say broadband, what I mean is that instead of measuring at just one frequency, like I mentioned before, we're measuring over a range of frequencies. So we're able to look at that target strength curve as a function of frequency. And I'm going to focus today on just one of the transducers on DeepC. There are 7 broadband transducers. This is called the AirMar, and it has a frequency range that spans 25 to 40 kilohertz. And I'll note that this frequency range is particularly interesting because it contains 38 kilohertz. The frequency that I mentioned is particularly relevant in fisheries acoustics. And I'm going to focus on data from one deployment of DeepC that took place in 2019 during a cruise off of the New England continental shelf break, shown here on this map. And I'd just like to acknowledge that all of the fieldwork for this project occurred before I started my postdoc, and unfortunately, due to everything happening in the last year, was not able to collect any data. So I'd just like to thank the folks who did. And this photo here is just a photo of deployment of DeepC. Just to give you a little bit of a sense of scale, it's a really big system. It's like the size of a car. Okay, so here you can see the shipboard echosounder data from the deployment that I'm going to focus on. And the white line shows you the depth of DeepC as a function of time. And all of this data was collected during the daytime. So this is during the period when our migrating organisms are back down in the mesopelagic. So we can zoom in on what the data from the AirMar transducer on DeepC looks like. So here you can see data collected around 600 meters deep, within the scattering layer around 500 meters deep, and around 300 meters deep. And as we hoped, with this close-up data, we can now see individual organisms. We're not measuring volume backscattering, where we're seeing many organisms within the beam at the same depth. We're able to make out individuals and measure their target strength. However, as you get farther from the transducer, we go back to measuring that volume backscattering, particularly in the more organism-dense areas. And so we're no longer able to make out individuals. So in order to pull out these individual targets that I can look at their target strengths, I implemented automatic target detection. So first, applying a threshold to determine where we can reliably detect individuals rather than multiple organisms. Detecting targets that are relatively on axis or in a straight line from the transducer. And then by taking an FFT and a narrow window around each detected target, we can calculate the target strength of the target in our frequency band, which you can see for this specific target here. So now I can pull out a bunch of targets, and I've got these target strength curves, and I've got a whole bunch of them. But what does that actually tell us? Well, we can turn to some acoustic scattering models to better understand that. So here you can see what the scattering from a fish with a swim bladder or any organism with a gas inclusion is going to look like. And I've highlighted here in yellow the AirMar frequency band. So these are the frequencies that we're measuring. And as I sort of walk through these models, think about what we're seeing in that frequency band and what we might expect to see in our measurements. So for a swim-bladdered fish, we see a steeply increasing slope that comes to a peak in a resonance frequency and then tapers off and eventually gets relatively flat at higher frequencies. And the characteristics of this curve are not only going to be a function of frequency, but also of the depth, as I mentioned before, the size of the fish in the swim bladder, the shape of the swim bladder, and the material properties of the fish. So we can take a little bit of a look at the dependence on some of these parameters. So if I hold everything else constant and make my, my swim bladder smaller, the resonance frequency is generally going to get higher in frequency, and our curve is going to get lower in target strength. And we see the opposite as we increase the size of that swim bladder. Again, if we hold everything else constant and move our fish deeper, we're going to see the target strength increase in frequency and we're going to see the resonance frequency increase in frequency and decrease as we move it shallower. Of course, this is going to be a little bit more complicated for a real fish. I'm not accounting for changes in shape and volume that might occur as the fish changes depth. This is just accounting for changes in pressure, but just gives you a little bit of a sense of the changes that we might expect to see. We can also look at what scattering looks like for a fluid-like organism, so something like a squid. And here we're going to see generally lower target strength and a less steep increasing curve as a function of frequency. And again, holding everything constant and making this a smaller squid, we generally see decreasing target strength. And as we make it bigger, we see increasing target strength. We also start to see more complex scattering at lower frequencies for larger organisms. Okay, so hopefully you are paying attention to that yellow band that I highlighted, because now we can take a look at what this means for our measurements. What shapes of target spectra do we expect to see? Well, we expect to see gas-bearing organisms that are at resonance, so their resonance peak is in the band that we're measuring. Below resonance, so their resonance peak is higher than the band that we're measuring. Above resonance, so their resonance peak is below the band that we're measuring. And fluid-like organisms, so these are generally going to have lower target strengths and in a less steep slope. And when we look at the data that we measured, we see just that. Most of the target spectra that we measured fall really well into these four classes. Of course, we're talking about data collected in the real ocean, so I'm going to add a fifth class here for target spectra that are complex or unknown. So these are not necessarily described well by the scattering models that I've presented here. So we can look at these target spectra and group these by hand, and we can start to do some analysis that way. You can see some preliminary results in a paper that Chris Bassett published last summer. But if we want to start to apply this sort of analysis to all of the data that we've collected with deep sea, we need a way to do this automatically. So there are a few different approaches we could take here. We could make hard and fast rules for if target strength is increasing by this much at this frequency or exceeds this frequency, it belongs to this category. But that's going to be difficult to tune and really susceptible to noise in the data. We could annotate a whole bunch of training data and use that to train a machine learning model. But there we're subject to biases for the target classes that we don't have very many of. We're just not going to have a lot of training data for them. So I was curious if I could train a classification model using model-generated training data. So use those scattering models that I just showed you to generate synthetic training data. In order to do this, I used parameters representative of what we know about mesopelagic organisms, generated some 5,000 model-generated target spectra in these four classes. smoothed the data to address noise in the real measurements, and calculated the first derivative because the measure of slope is going to be particularly relevant here. And then fed this all into a really quite simple machine learning algorithm, k-nearest neighbors, and used that to predict which class each target spectra belonged to. And of course, this doesn't capture those complex target spectra that our models don't describe well. So I applied a distance threshold to assign a class of complex to any of the target spectra that didn't fit well into these models. And in order to see how well this performed, I annotated 400 target spectra by hand that were randomly selected. And overall, 91% of the classifications agreed with the hand annotations. So this is overall a pretty promising statistic. And when you dig into the results further, where the model and the annotations didn't agree were generally cases where the human annotation was not straightforward. So these might be cases where we have a resonance peak right on the end of the band, and it's kind of difficult to decide if it's below resonance or at resonance, or where the data is particularly noisy and it was difficult to assign a category. So I would argue that this machine learning approach is, if anything, at least more consistent than human annotation. Okay, so now we have a tool to analyze these target strength measurements. What does that actually tell us about the data from this deployment of DeepC? Well, here you can see the fraction of targets that were at resonance, below resonance, above resonance, fluid-like, and complex or unknown as a function of depth. And we can draw a couple of conclusions by looking at this. First, we see very few complex and unknown target spectra. So this is telling us that overall, these scattering models did a pretty good job of describing what we're seeing. We also see relatively few fluid-like organisms. Though I'd like to point out, this doesn't mean that there are not fluid-like organisms in the mesopelagic. It means that they are weak scatterers that we're not necessarily measuring very well in this frequency band. So they aren't contributing to the backscattering that we're seeing, for example, at 38 kilohertz from the surface. Finally, we can look at this and see that it doesn't exactly match what we would expect given the depth dependence of scattering. So this tells us that in addition to these changes that we see as a function of depth, we're also seeing changing species compositions throughout the water column. And we can also look at the target strength specifically at 38 kilohertz, that frequency that I said we're particularly interested in for comparing with other studies, and how that changes as a function of depth. So you can see that for each one of our 5 classes here. And overall, you can see that the organisms that are at resonance have the highest target strength. This makes sense, right? We have that peak. And the fluid-like organisms, as we would expect, have the lowest target strength. But looking at this begs the question, okay, what does this mean for our measurements from the surface? How much volume backscattering comes from organisms that are at resonance, below resonance, etc.? So we can estimate that. And so on the left here is that same plot that I just showed you. And on the right here is the estimated fraction of volume backscattering at 38 kilohertz that each one of those categories contributes. So you can see that the shapes of the curves are quite differently here. Specifically, we're seeing a disproportionately high contribution from organisms that are at resonance. Right in the middle of our scattering layer, around 450 meters, organisms that are below resonance make up about 67% of targets, and organisms that are at resonance make up only about 27% of targets. But the below resonance organisms contribute only about 33% of volume backscattering, while organisms that are at resonance contribute 66%. We can also look at this and see that gas-bearing targets account for nearly 100% of volume backscattering. And in general, this is good news because this is an assumption that's been made in a lot of previous studies looking at shipboard data. And they've made the assumption that what they're measuring is entirely contributed by organisms with gas inclusions, like fish with swim bladders. So this is kind of confirming that that is a good assumption to make here. So hopefully this gave you a little bit of a taste of how we're starting to learn more about organism distributions and scattering in the mesopelagic using the broadband echosounders, and hopefully this information can start to inform how we're making measurements from the surface. But I'm going to shift gears here a little bit to talk about a different subsystem on deep sea, the holographic camera. And so the holographic camera on deep sea is up front here. And a holographic camera or a holographic microscope looks something like this. So there are two housings, a laser and a camera, and a laser shines between the two. And the region of water illuminated is generally referred to as the imaging volume, because any particles within that volume will diffract the light, and the camera records the resulting diffraction pattern in what we call a hologram. So that hologram is going to look something like this. We can zoom in on one of these diffraction patterns here. So you can see how from looking at this, we can tell where in the XY plane our targets are, but we can't necessarily tell what they are, how big they are, or where in depth, sort of in the Z direction, this target is in space. But by back propagating the diffraction pattern to the depth within the imaging volume where the target is, we can reconstruct a pretty high resolution, clear image of the target. So here you can see that that was the diffraction pattern of a copepod, which is a type of zooplankton that is particularly prevalent in the mesopelagic. And I'll just sort of point out here, now I'm talking about an entirely different size scale of organisms that we were looking at with looking at acoustic scattering at 38 kilohertz. Before we were looking at fish, now we're starting to look at millimeter and micro scale organisms and marine snow and things that you might not be able to see well with the human eye. So, of course, if we perform the reconstruction at the wrong depth, so I instead do my reconstruction at this point here, we don't see the target and we aren't able to classify it. So there are a few reasons why we want to use holography as a tool to look at plankton and other particles in the ocean. Compared to other optical imaging systems that are able to look at organisms of this scale, we have a pretty large, well-defined imaging volume. And so this is especially advantageous in areas where particle density is relatively low. We're able to just image more water, see more particles than we would be with some comparable systems. The focusing for the system is also done in post-processing. So this means that the hardware is actually pretty simple. We don't have any complicated lenses, and we aren't missing any particles that aren't in focus. So just kind of back to our rendering of deep sea here. As I mentioned, the holographic camera is up front. And just to orient you a little bit, the imaging volume looks something like this. So the separation between the laser and camera housings is about 1 meter. And where the edges of that square, this is not at all to scale, are like 6 and 11 millimeters, sorry. And I'll just note that compared to a lot of other holographic cameras that have been used in the ocean, this is a pretty large imaging volume. We have a separation distance of 1 meter, but a lot of comparable systems have imaging volumes on the order of just a few centimeters or even shorter in length. So we're imaging a lot more water with this system. So when I started looking at the data from this holographic camera that had been collected by Deep Sea, I got really excited. I had these big dreams of building a neural network to classify copepods and estimating copepod abundance and comparing that with the high frequency acoustics on deep sea. But I pretty quickly realized that I was about 20 steps ahead of myself because actually extracting those targets from the data to do that machine learning step and start to draw conclusions from this data is pretty challenging. And there are a few different reasons for this. The first is that performing reconstruction is really computationally expensive. It requires calculating the FFT and the IFFT of the whole hologram, which is some 3,000 by 4,000 pixels large. And on top of that, if we store those reconstructions for later analysis, we're creating a whole nother image of that size with every reconstruction that we calculate, and just producing more and more data that we need to handle. Second, the position of the target isn't known beforehand. So we need to determine where the target is in focus, either by hand or using some sort of focus metric that tells us where that target is clearest. And so this is inevitably going to require calculating a lot of reconstructions, which is computationally expensive. Of course, I'm not the first person to come across this problem, and there are a few existing methods that have been developed to do this target extraction and classification automatically. The first is to perform high resolution reconstruction of the hologram, perform target detection on that reconstruction, and pull out the targets from that sort of three-dimensional reconstructed volume. There are a bunch of variations on this approach in the literature using different target detection schemes, different metrics for determining where a target is in focus. And overall, they can give really accurate results and don't require humans to pull out where those targets are. But it's still really computationally expensive with that reconstruction step. And it starts to not be practical when you have long time series data or when the imaging volume is larger, and we have both of those problems. The second approach, and this is from a paper that was published just at the end of 2020 in December, is to perform classification on the diffraction pattern in the raw hologram. So just bypass that reconstruction step altogether. And you can see, I pulled a figure from that paper here. And if you look at number 5 here, when we're looking at the diffraction pattern, you can see that it looks quite a bit like a copepod. We see the body and we see the antenna. You can kind of wrap your mind around how we're able to do automatic classification of that diffraction pattern. And this is generally going to be true when we have small imaging volumes. When targets are very close to the camera, the diffraction patterns are going to more resemble that target. But when they're farther from the camera, this isn't going to work so well. You'll recall the copepod that I showed you on the first slide. Its diffraction pattern looks like this, and it does not look anything like a copepod. We don't see the body, we don't see the antenna. We don't really have much of a hope of classifying it without reconstruction. So I realized I needed a different approach to be able to process these targets, to be able to process a meaningful amount of the data that we have. So I developed a processing pipeline to rapidly extract the focused targets from the holograms without requiring reconstruction of the full hologram. This pipeline has 3 steps: select holograms for processing, detect the diffraction patterns, and then perform an optimized reconstruction scheme to focus those targets. So first, selecting holograms for processing. In the data collected by deep sea, particularly close to the surface, we had quite a few holograms where optical turbulence interfered with the image, and this created problems for automatic target detection or focusing algorithms. So I wanted to remove these from the processing pipeline. So to do this, I used a transfer learning technique. I used a pre-trained neural network to extract features from each hologram, and then fed those features into a support vector machine for classification. And after testing this on hand annotated data, it had a 94% precision and 91% recall. So overall, I was pretty confident that this is going to remove those holograms that are going to bog down our automatic processing and return inaccurate results. The next step is to detect diffraction patterns of targets in the raw hologram. So to do this, I first subtract the background, then use a low-pass filter to suppress background noise, apply a threshold to create a binary image, then apply a series of morphological filters to suppress noise further and bring together any nearby components. And then from this processed image, we can now detect where in this raw hologram we have diffraction patterns that are associated with targets. So, of course, I've chosen a nice, pretty example for my seminar here today, but I wanted to get a better idea of how well this target detection scheme worked. So I needed some sort of ground truth for how many targets were present. So to do this, I implemented one of those methods that I told you about earlier and performed target detection in full high resolution reconstructions of each hologram. And I implemented this for 271 holograms to generate sort of a ground truth data set for where I have targets. And I'll just note that this ground truth method was really slow. On my relatively powerful desktop computer, this took over 20 minutes per hologram. Of course, this could be sped up on a different computer or using a cluster or something like that. But I'm just going to give you some processing times for the sake of comparison. Okay, so how well did the diffraction pattern detection on the raw hologram work? Here on the x-axis, you can see the size of each target in the reconstruction. And on the y-axis, you can see the fraction of targets in that size range that were detected based on their diffraction pattern in the raw hologram. And while our detection capabilities roll off quite a bit for smaller targets, overall, 98% of targets larger than 0.1 millimeters squared were detected. And in general, that size class is going to include most of the targets that we're really interested in, the copepods, larger zooplankton, and larger pieces of marine snow. We can also look at our precision. So looking at how many of those diffraction patterns aligned with where I had a ground truth target, had 94% precision, or only 6% of detections were false positives. We can kind of shift the trade-off between precision and detection accuracy here by changing some of the detection parameters. But overall, for the targets that I was interested in, I was pretty happy with this trade-off here. So this brings me to the last step in the processing pipeline, optimized reconstruction. So here you can see a reconstructed target from this hologram here. And this is just showing you here the bounding box of that target that I detected. And calculating this reconstruction of this whole hologram to get this little target took about 1.3 seconds just for this one reconstruction at the depth of the target, again, on my desktop computer GPU. So looking at this, I was kind of wondering, this is a really small part of this hologram that where this target is. What if I reduce the amount of image that I'm calculating the FFT of when I do my reconstruction? So if I just use the information within this bounding box, we calculate the reconstruction really quite quickly, but if this reconstruction here, we lose a lot of information in detail that we had in our reconstruction. We can't really tell it it's a copepod or assign this a class. But as we start to increase that window just a little bit, we're still over 100 times faster than calculating the full reconstruction, and we're getting a starting to get a clearer image. And as we increase the size of that bounding box, by the time we're adding 4 millimeters to each side of the target, it's 21 times faster to compute, and our reconstruction is really just about as clear as it is using the full reconstruction. So this is great to cut down on the time for reconstruction. We're able to calculate them much more quickly, but we still don't have a way to automatically determine what the z-value for this reconstruction is. So to do this, we need a focus metric, and I settled on this one, the standard deviation correlation function, which in this 2016 paper was found to have a pretty good trade-off between performance and computation time. And you can just see this metric calculated at 1 millimeter intervals for that target that I just showed you. And right here at this pretty sharp peak here is in fact the point where our target is in focus. So now we can calculate reconstructions at different depths in the imaging volume, calculate this metric for each one of them, and call the reconstruction with the maximum value in focus. And this still represents significant gains over reconstructing the full hologram, but it still means calculating a whole bunch of reconstructions that we're just going to throw away. So to further speed this up, I implemented the golden section search, a pretty simple search algorithm to speed up how quickly we're able to find that peak. And doing so cut down the number of reconstructions that we need from 1,000 to 31 in order to get 1 millimeter precision on the depth of focus. So I wanted to see both how reliable the focus metric is and how well the optimized search algorithm works. So for 321 targets, I both calculated those 1,000 reconstructions at 1 millimeter intervals and calculated the focus metric, and also implemented the golden section search method and looked at what this approach had called the focused reconstruction and determined whether it was in focus, sufficiently focused, so maybe a little bit blurry, but I could still assign a coarse classification, or out of focus. And overall, 88% of targets were sufficiently focused. So this is a pretty promising statistic. Obviously, there's room for improvement. But I'd like to point out that something that's really nice about this is that we've quantified how well this works at every step along the way. So when we start to use this method to extract targets, we can understand the implications of our results as we start to sort of draw quantitative conclusions. So finally, of course, I had to pull all these steps together and process some holograms. So I implemented this three-step process for about 500 holograms. And just some statistics on how well this worked, pulled out an average of 3 targets per hologram. And overall, 83% of those targets were in focus. This is a little bit lower than the previous slide because now I have compounding errors at each step in the processing pipeline. But this still represents way more data than we've been able to extract before. We now have a large data set that we can process more holograms and grow this data set to start to get to those steps of doing automatic classification, estimating abundance, and starting to do some science with this system. And the best part is that this took, again, on the same computer, an average of 8 seconds per hologram. So obviously varied with the number and size of targets, but represents huge gains over what we were able to do before. And it starts to become feasible to process the amount of data that we've collected with DeepC. So finally, just a couple of big picture conclusions. Hopefully I've shown you today both why exploring the mesopelagic is important and why it's quite challenging. But with instrumentation systems like DeepC, we're starting to collect rich, new, exciting data sets. However, before we can start to use those data to answer the big science questions that we have, we need to develop the tools to quantitatively and accurately process those data. And so I've shown you some steps that we've been able to make in that direction today. And so with that, I'd like to thank my collaborators on this work and the funding sources and open it up to questions.
That was wonderful. Thank you, Dr. Cotter. If anyone has any questions, you can feel free to write it in the chat or unmute yourself and directly ask. And we actually have a question written in the chat right now, so I'll read it aloud. This is from Liz Widener. She said, "Great presentation, Emma. Really wonderful. I'm wondering why you only looked at on-axis targets and sampling the targets. I imagine including off-axis data would provide a lot more information if you could correct for the beam angle."
That's a great point, and definitely something worth looking into. The system on DeepC is a bistatic system, so we don't have that beam correction super well defined more than about 2 degrees out. But getting better measurements of that is definitely something that I think is going to happen moving forward. So
I have a question. So talking about the broadband acoustics, and you have your classification models. Do you have any plans for the future on using the holographic camera to sort of ground truth those models from the broadband acoustics?
So the organisms that we're looking at with the holographic camera are zooplankton for the most part, and you would look at those with much higher frequencies than you would look at with fish, just because of where their scattering falls. But we do have some of those higher frequency systems on deep sea. At this point, I'm not working on this project anymore because I've moved on to a different position, but that's definitely kind of what I hopefully have been setting up for people to keep working on.
Right. Awesome. And so can I ask, like, what are your plans for working at PNNL?
Yeah, so I'm shifting gears quite a bit and working on marine renewable energy. I'm really interested in working on how we can use marine energy to power ocean observing, to just get more information and longer deployments than we've been able to previously. And I'm still fleshing out the answer to that question. I've been there for about a week, so.
Right, that's awesome, yeah. Thank you. Are there any other questions? We also have someone that wrote, "Thanks, Emma, great talk." I have to run, so he had to leave in the middle of the meeting, but he said it was a great talk.
Yeah, that was from Tom Liebman. I have a question. I believe you might say that, and maybe I just missed it during your talk. Can you say again how you reconstructed the hologram? Is it just a Fourier transform that you ran on the image, or?
Yeah, so the reconstruction, there are a few different algorithms that can be used for reconstruction, but in general, sort of the fundamental theory is that you take the FFT, so transform it to frequency space, propagate that frequency information to the depth of the target, and then go back to the time domain, so calculate the IFFT.
So for that, you needed to know the location of the organism within this volume, and this is why you did that autocorrelation to find it.
Yeah, so you can calculate the reconstruction at any depth, but your target's not going to be in focus at the depth where it's not. So you can calculate the reconstruction and essentially create a 3D volume that contains all of your targets. And then at the depth where the target was is where you're going to get that crisp image.
Okay, and just to clarify, when you say depths in the optical connotation, it's not water depths as in acoustic.
Yeah, sorry if that was
from the that's the distance
sorry
from
if that
the
was
camera from the
confusing. Switching the implications
the optical and the acoustics. Yeah.
But yes, there I mean depth within the imaging volume, so 0 to 1 meters.
Yeah. Okay.
We have a couple more questions. So Julia Chawarski said, "Really great talk, Emma. I'm working on this exact same stuff for my PhD and appreciate the work going into it. How do you determine the correct window size for FFT for acoustic targets, or do you vary your window size based on some other parameter?"
So there I set it kind of thinking about the largest targets that we expected to see, and looked a little bit at dependence of the results on how I changed the FFT size. I think I ended up using 30 centimeters and did try varying that, and you're going to get more noise or potentially more nearby targets as you increase it. And as you decrease it, you're going to not have all of the information. So it is a pretty sensitive trade-off and going to be different depending on what environment you're looking in.
And then we have a question from Derek Sowers. Are you aware of other vehicles similar in capabilities to deep sea, or is it essentially one of a kind? Are there changes you would recommend to further optimize a tool like this for exploration of this kind?
That's a good question. There are similar systems being developed. There's a group out of Norway that has something similar. I think deep sea is kind of the most the largest with the most instrumentation, which is both a blessing and a curse. We have all of this data, but it's also really difficult to deploy, and you can only do it from some vessels, and it's pretty big undertaking to get it to sea. And changes to further optimize it. I think we're learning a lot about what the instruments that are most useful are. So for example, there's a multi-beam sonar on deep sea, and I spent a while looking at that data. And this isn't to say it couldn't be useful, but just figuring out, looking at the data, it looked really rich and interesting, but figuring out how to get anything quantitative out of it was not immediately apparent, and is still a bit of an open question. And so I think in future iterations, thinking about what those sensors that are most powerful, maybe downsizing it a bit to just have some of those.
Great, thank you. Are there any other questions? Okay, so it seems like we are done with questions and it's 4 o'clock. So thank you again, Dr. Cotter, so much for spending the time to present for us. We really appreciate it. And yeah, and then our next seminar will be next Friday. So we are off this coming Friday. We have another seminar next Friday. So we hope to see you all there. Thank you again, Dr. Cotter.
Bye-bye.
Bye.